All right, we're going to be doing a lab here, the Candy DNA Replication Lab. So we've been investigating DNA and its shape, the twisted ladder, and how we have nucleotides, the 4 nucleotides, adenine, thymine, cytosine, guanine, how they match complementary to each other. And so yesterday we learned how DNA replicates itself. Basically 3 steps: DNA unzips, and then the nucleotides come in and match complementary to each template. And then you end up with 2 identical DNA molecules.
Okay, I will get that in for you.
So we're just going to take a look at a fun little candy lab that goes through this. So the materials that you're going to need: 4 toothpicks. I guess I can't count, but 4 toothpicks, 2 pieces of red licorice. I need to add 2 pieces of brown licorice. 4 yellow gummy bears that will serve as our adenine. 4 orange gummy bears. You see 3 there for thymine. 4 green for cytosine, and 4 red or pink for guanine. So our gummy bears are going to represent our nucleotides. So our directions, it says, use your 2 red pieces of licorice to assemble a strand of DNA with the following nucleotide bases: A, T, C, and G. So A is denoted by a yellow gummy bear. Okay, so here's my A. And then our second nucleotide is, oops, I covered it up. T, thymine. Thymine's going to be orange. Okay, and then our third nucleotide is cytosine, that's green. And then your red or your pink gummy. And we're gonna take licorice, and this is gonna represent 1/2 of our DNA. So what you get to do now is take your toothpicks, because they're gonna help hold our nucleotides together, and I guess you're going to, for lack of a better term, but stab or impale your gummy bears. So. Sorry, Mr. Gummy Bear. Okay, so I have 1/2 of my DNA strand. So that's step 1, okay? So our toothpicks will attach the nitrogen bases to the sugar phosphate backbone, which represents our licorice, okay? Now step 2, attach the complementary nucleotides to the other side, and we're gonna match complementary. So I got my other licorice here, but now I need to match complementary. So remember, adenine is gonna pair with thymine. So yellow gummy bear is gonna pair with orange gummy bear. All right, I'm gonna attach the sugar phosphate bone last. So orange pairs with yellow. And then green, cytosine, pairs with guanine, so green's gonna pair with our pink or red gummy. Oops. Stab these a little bit more here. Okay, now I'm gonna close it up, the other half of my licorice. See, sorry, bumping the table. I have constructed somewhat of a DNA model here using gummy bears, licorice, and toothpicks. Okay, now like any good science, you should probably start recording what you've done. So it says carefully color code and label your original strand, step 3 here, in the space provided using all the labels used in the directions on the observations table. So that's on right here. So we just constructed our parent DNA, and we need to label and color, okay? So I'm gonna use black here. Our yellow gummy bear is what nucleotide? Adenine. So I'm just gonna represent adenine with the letter A. And A pairs with T, okay? And if I look at my model, T pairs with A. My next gummy bear down is green, and green is cytosine. Why they didn't make that guanine, I don't know. Okay, this is my sugar phosphate backbone. We'll just say sugar phosphate. I guess I would also accept like the backbone. Maybe you want to label that the backbone, right? And now we got to color this. So the sides of my letter are red. Take a picture and put it on Google Classroom. I'm doing a lab for those that can't do our lab tomorrow.
Wait, I lost a lot of my papers at home, so I'm just gonna have to redo them.
Okay. So T's are orange, A's are yellow. Unfortunate timing. Okay. I guess I could have used pink for that too, but it doesn't matter. Okay, so we just did step 3. Step 4, using scissors, separate the 2 strands by cutting down the middle, okay? So remember in DNA replication, okay, the DNA strands unzip, so we have to cut through our toothpicks so we can match complementary to the inside strands. Okay, so we're gonna use scissors, we're gonna separate it. This portion of the lab will most likely not be done, but with your partner, I want you to think about, actually, we're just gonna do it. Okay, so hopefully these scissors can cut through. Cutting through, looking at through a camera. Hey, this is not bad. I don't know why during the lab, whenever we get to this part, it always seems difficult. Okay, so our strands unzip, okay? Whoop. And now our second step is that these nucleotides, they're gonna come in and they're gonna match complementary. So yellow pairs with orange. Okay. Orange pairs with yellow. There's my A and my T's are done. Then we got our Christmas colors, cytosine pairs with guanine. And guanine pairs with cytosine. So there's 1/2. Now we gotta do the other half. So there we go. Almost there. Oh, of course you would fall off. That's a messed up gummy bear. His leg's all messed up. Okay, so my nucleotides match complementary, but then to show that this DNA molecule has undergone replication, I'm gonna stick a different color sugar phosphate backbone to show that each one of these original strands, like 1/2 of it is from the original strand, and the other half of this new DNA molecule is got a new backbone, okay? So now, gotta go back to, get this out of the way, put this on the plate. Going back to our coloring, gotta color what we see, okay? Stick that off to the side. Okay, so I'm gonna color the outsides red, okay? Because those are the original strands. That licorice was from the parent strand, so this means over here, I'm gonna color red. All right, and our DNA sequence is gonna be the same. A pairs with T, then it was T, C, G. The new sugar phosphate colors, we're gonna color, you can color brown or black, because I have chocolate licorice, not black licorice. A lot of kids don't like black licorice. And then I'm gonna go ahead and just take a look. It's gonna be yellow, orange across. So my T's are orange. My A's were yellow. My C's are green. And my reds are guanine, or pink. Okay, so we just did replication of DNA, and you can see that it is the original. Our new copies are exactly like the original. The only thing different is, you know, the sugar phosphate backbones here. So we got some questions that ask about how are the new strands similar? How are they different? And now this is a model of DNA replication. And so what you need to do is tell me what each thing, what was represented by our model. So for adenine, we used yellow gummy bears to show adenine, okay? For guanine, I believe it was the red gummy bear. Now you should be able to get all of these. Toothpicks might be difficult, but toothpicks are like the bonds that hold the nucleotides together. And I believe they say that. Maybe they don't, but toothpicks are bonds. We haven't talked about helicase, so really we're just gonna cross it out. But if you really, really wanted to know, it was the scissors. Helicase is the enzyme that unzips DNA. And DNA polymerase is the thing that matches complementary to it, and so that would be you. You're the one that was matching all of the gummy bears. But again, we're just gonna cross that out. I don't want to worry about this, but the rest, the gummy berries you should, and the red licorice, what that represents. Okay, and then one more question, why is DNA replication necessary for cells? All right, so that's the lab. So you just follow along and complete the lab.